Welcome to the IBDC Seminar Series 2026. So basically, this is a seminar series which was designed in a way to develop content for the users so that they can be able to use our resources seamlessly. So today's webinar is on Indian Animal Phenome Database, which is a newly launched database which is dedicated for animal phenotype data. And as per the webinar or the agenda goes like initially we will welcome about the series and then we will go to the information about, or you can say the overview about IBDC and its researches. And then we will describe about the Indian Animal Phenome Database and why this database is important and why exactly the phenome data is crucial for the animal science advancements. And lastly, we will go through the demo data submission in this database. So coming to the first, let me introduce to my team, the Indian Animal Phenome Database team, as I'm Dr. Sonia Balan. And together with Dr. Nivedita Yadav, Ms. Isha Singhi, and Dr. Vibha Abrar, we are the team behind the development of Indian Animal Phenome Database. So I request you all, all to please mute your microphones and also use chat for the question at the end of the webinar. This webinar will be recorded for the future references. And if you have any questions or queries regarding the data submission or some other issue, you can always write to IPR support at ibdc.rcb.is.in. So to start with, we are beginning with the part one of this webinar, which is on overview of IBDC and its services. So for that, I invite Dr. Nivedita Yadav to brief you about the IBDC and its services and what all databases it hosts. So Nivedita, over to you.
Yes, ma'am. Thank you for introduction. And very good evening to everyone and welcome again to this webinar. We are very hopeful that you will get something very interesting by the end of this webinar. So stay with us. Ma'am, could you please unshare your screen so that I can? Yes. Before moving towards overview of IAPD, just let me introduce about IBDC and its services. This slide, I will explain and make you understand that the landscape before IBDC was established. We all know that biological data is humongously diverse. We generate data related to gene genome variations, functional genomics, proteomics, and molecular structures, metabolomics, phenotypes, and image-related data. So each experiment produced different kind of data in different form, like metadata files, ontology, raw data, process data, and many more kind of this. But what happened to many of these data? We all know that all these data is scattered and some stayed in personal computers, some in lab services, some in external drives, and some simply forgotten and somehow it lost and never reused. Most of the researchers are submitted data only when it requires to publication. And where did we submit it? It is generally international repositories like EMBL, GenBank, PDB, ArrayExpress, which creates a dependency to external fragmented and ecosystem. What it results? This results no centralized repository, limited interoperability, fragmented data management, and external infrastructure dependency. In short, we are operating in a fragmented and external dependence ecosystem. So, and also naturally, one more thing is also in researcher, a thought, why should I spend time in submitting data where you can utilize this time in generating more data? But the real question is, can this scientific progress truly accelerate if data is not structured, shared, or used? This challenge led to a creation to a national solution. And this is what IBDC enters in the pictures. Now let us see what changed after IBDC is established. Instead of scattered data, now we are having a structured national data submission system, which is IBDC itself. IBDC provides infrastructure layered for biological data management in India. It provides different type of biological data system services, and having data sets like gene, genome, proteome, metabolomics, and many more. And now we are one coordinated ecosystem, that is Indian Biological Data Center. Now data does not remain lost in individual lab, it moves into India's national repository. This repository follows a FAIR principle. FAIR stands for findable, accessible, interoperable, and reusable. This means data is not just stored, it is structured, standardized, and ready to use for other scientific community. By having IBDC, India is now supporting digital sovereignty in biological data. At the same time, IBDC ensure interoperability and global databases. So we are connected globally while building strength to our nation. Primary databases like GeneBank, EMBL, PDB, NCBI, this is also in the limelight. But now where India is in the queue also, and India also have its own national repository, and this ecosystem working alongside with them. This major steps contains data-driven decisions and self-reliance in biological data. IBDC is not just a repository now, it is a foundation of future biological research in India. This is how we move towards a strong, more connected, self-reliant scientific ecosystem. So let me give you a brief introduction about IBDC. So IBDC is India's first lifetime data repository. It is supported by Government of India through Department of Biotechnology, and it enables biotech guidelines, which promotes responsible data sharing. It is a joint project of Regional Center of Biotechnology, National Institute of Immunology, International Center for Genetic Engineering and Biotechnology, and National Informatics Center. IBDC is located at Regional Center of Biotechnology itself, which is an institute of academic institute and institute of Government of Biotechnology, and also collaborating with programs with UNESCO International Category Center 2. IBDC also have one data recovery site, which is in Bhubaneswar, where real-time data exchange happens for the recovery purpose. Also, in 2022, IBDC was dedicated to the nation by Dr. Jitendra Singh, Minister of Science and Technology. Let us know about the IBDC infrastructure. One of the most important facility of IBDC is its high performance computing system, named as BRAHM. BRAHM HPC has a huge capacity of storage, that is 5.5 petabyte, computing power 961 teraflops, which means it can store many large files, and also it facilitate high power computing. Because of this facility, IBDC and researchers are performing many advanced studies like genome analysis, RNA sequencing, disease-related gene expressions, and other biological analysis. Currently, HPC supports 128 faculty members and 62 institutes across India, helping them managing their research efficiently. IBDC also provide a vision to expand the infrastructure by up to 10 petabytes, where 7 petabyte could be used for IBDC and 3 petabytes at DSR. Now let us understand the bigger picture, the mission of IBDC. IBDC enables fair and interoperable biological data to support data-driven research, innovation, and decision-making across agriculture, health, and sustainability domain. What does it mean in a simple word? It means data generated in India should be structured, connected, reusable, and useful for national growth. If we talk about IAPD, with a simple example, the ecosystem especially contain plant, agriculture, microbes, livestock, and human. And all these domain handles many kind of data, like I have explained in past slides, like gene, genome, RNA, protein, and many more. All these kind of data flows to IBDC as a centralized system, built a FAIR principle, to get a impactful output. Like in agriculture, uh, climate resilient crop innovation, it enables faster evidence-based diagnostics, and it helps in health productivity. So the impact goes beyond the data management. It direct support real solutions to the world. Now the question is, what IBDC actually do? IBDC basically provide some services like biological data archive solutions, high performance computing facilities, support to projects of national importance, big data analysis, user resources and documentation, training of data submission and analysis. So whether you are a researcher generating data or scientist analyzing a large data set, IBDC supports you as every step. Yes, now let's talk about a very important concept, that is fair data principle. FAIR stands for findable, accessible, reusable, interoperable, and reusable. It means your data should be easy to find, easy to access, easy to combine, and with other data, and also easy to use in future studies. The answer is very simple, because it is directly benefit to researcher. It helps you to provide visibility, increase citation of your work, gain recognition, and ensure reproducibility of your result. When your data is fair, your data follows FAIR principle. It moves just not being a data, but become a knowledge. And that knowledge can be reused again and again and can help in discovery. Research is now just, I'm giving you one example through this cycle. First, researchers start the plan, get the data generated, collect the generation data, process it, analyze it. Now, one way is to publish the result and forget about the results and data. And one wise option is also it to reserve and save the data by submitting it to IBDC, so that IBDC could give them a data accession number, and that could be a national accession, and that accession can help them to trace their project, and also to give a global visibility. Now we can think science is not just about generating data, it is also making data usable, reusable, and impactful. Now that you know about the IBDC, let me show you the data submission and services ecosystem of IBDC. This ecosystem explains how different kind of biological data are collected, stored, and managed through the IBDC platform. IBDC work with many types of data such as gene, genome, RNA, protein, molecular structure, chemical, and phenotype. For example, data related to gene, genome, and RNA are submitted to INDA, INDACA, and Indian Array Data Archive. For protein and structure related data, we have platforms such as Indian Proteome Data Bank and Indian Structure Data Archive. These platforms help researchers to store and access protein related information very easily. For data related to chemical and small molecule, IBDC provide Indian Metabolome Data Archive, which support metabolic research. For biological image data, IBDC also have Indian Biological Image Archive, where researcher can submit and explore image-based data. When we talk about phenotypic data, it is observable characteristics of plant, animal, microbes, and human. We have Indian Phenome Repository. Under this umbrella, we have Indian Crop Phenome Database and Indian Animal Phenome Database that we will explain in next slides. And future also, under this umbrella, we have a vision to add Indian microorganism and Indian human phenome database. Along with data repository, IBDC also provides services platform that help users to analyze their data. One such platform is BioNode, and one is Bioverse, which is under development. So stay tuned with us. Some more portals are also in upcoming phase, that is Indian GWAS repository and literature database and genome assisted knowledge discovery platform. So IBDC is not just a place to store data. It is a complete ecosystem where data is collected carefully, analyzed, and finally to support new researcher to the discoveries. This is we have currently 4 portals. So for getting more information about it, I would request you all to go to the website of IBDC and explore all the data portals. IBDC also handles some national importance mission projects under nucleotide and crop. These are the list, and some famous and major projects are Genome India, INSACOG, and Indian Human Microbe. And in crops also, we have the list of major projects. This is the current statistics of IBDC. Currently, we have exceeded more than 1,000 terabyte of data site and which is submitted. And this stat shows in how much it giving services to a research community in this short span. Along with all these are our extra curriculum. We have successfully conducted 3 international conferences, and in future also we will continue it. So now it's time to understand animal phenome data, and over to you, ma'am. Hope this introduction helps everyone.
Thank you so much, Nivedita. Now just let me allow to share my screen.
Yes, ma'am.
So now coming to understanding the animal phenome data and introducing the Indian Animal Phenotype Database to all of you. So coming to this, to start with the introduction about the IAPD, it's important to give a brief introduction about what exactly and how important is the animal phenotype. So basically, when you say phenotype, it is basically the measurable physical or physiological or behavioral and biochemical characteristic of an animal resulting from the interaction between its genotype, environment, their interaction, and other genetic effects. So basically, you can say that when we say that genome is exactly, you can say the blueprint. You can say that phenotype is the actual performance or the result of that blueprint. But phenotype alone is not regulated. It is just from the genotype. It also influenced by the effect of the environment and genotype interaction, and also epigenetic effects. So when we say phenotype can be from simple traits like production traits, reproductive traits, environmental adaptability to the high throughput phenomics, which is sensor-based continuous phenotyping or the welfare phenotyping. So the key message here is when we say the phenotype, it is the functional expression of the biology, and it is the foundation of the very. So when we say that we want to improve the breeding program, we want to engineer the disease resistant livestock and the welfare, and ultimately towards the One Health system, which says that both all the animal, plant, and the human life altogether they are connected and they are interrelated. So when we think of human health, it's very important that we simultaneously involve or advance the animal health also. So this shows why exactly the phenotype data is important, because it laid the very foundation of genomic selection. So without a good phenotypic data, it is not possible to train the genomic prediction models or to identify the superior breeds, etc. So also it is very important for disease surveillance and One Health, because it allows you to detect early the zoonotic outbreak. It allows you to study the host pathogen interactions. It allows you to study the antimicrobial resistant monitoring in animal systems before it jump up to the humans. It also allows human welfare, welfare and the ethical feeding. Also, it is important when we say that we want to design the climate resistance, resilience, and sustainability of different aspects of animal sites. It's important for conservation and biodiversity, and ultimately, when we say that AI will definitely solve many of our uh, challenges in biological systems, and when we are moving towards precision livestock, it's important that we have good quality phenotypic data at a place and in a standard format so that it can be used for the precision livestock and AI. Basically, building models for the big data analytics and predicting something for better traits and better productivity of the livestock. So phenotype data is not merely just the measurement; they are the backbone of genomic selection, the bridge between the genotype and environment, and set the foundation for the One Health surveillance and drive the climatic resilience and the metric of the animal welfare and the evidence-based sustainable livestock policy. So when we see that what exactly are the scenario here in the global as well as in Indian ecosystem, when we think about the phenotype data archival, when you look at the global picture, you will find databases like animal KPL databases, CoreDB, which basically are dealing with KTL trait interactions, and we have FAN, which is basically the part of the functional annotation of animal genomes, and we have some uh, 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 online Mendelian inheritance in animals, which is catalog of inherited disorders of more than 500 species, which link their phenotypic descriptions to the gene mutations. But we also have model-specific phenotypic, very good phenotypic databases like mouse phenome database, which is very important and derives the majority of the work on designing the therapeutics in model systems. Then coming to the picture here, or you can say the animal genome data landscape in India, because as of now we don't have our repository, which we can say that we have a single place in India which is responsible for archiving all the data, and IBDC is just started to play that role for India and building different databases for the sustainable archival of the different biological data types. So when we think about what exactly the infrastructure have here in Indian research, which generate these animal phenotype data, or you can say the livestock phenotype data, we all know that ICR and many of the state agricultural university they generate a lot of deep performance report, field phenotyping. NDDB generate a lot of daily performance and productivity data. We have uh, large amount of population level phenotypic indicators. We have databases from NDAGR, which have the data of breed registrations and characterization records. We also have FAO linked Indian entries in GAD-IS, but all these databases are not dedicatedly for recording the phenotypic observations, and many of the big projects on genomics also, they generate a lot of phenotype data to study the association between the phenotype or the specific traits with the genotype, but most of them are project based. They store their data locally and not accessioned nationally. They lack standardized formats, and which limits its usability and cross-study integrations. As you can see here from the different stats which I collected from my search page, you can see that there are many of the institutions which are generating lots of animal data, but the irony here is we currently that all of the plant, or you can say that animal data is distributed in institutional silos, and uh, it's there within the institutions or within the labs. It was stored locally, and there is no unified national phenotype repository in India. When there is no such repository till now, the data sharing mechanisms are often informal and project specific. Long-term preservation and accessions are also inconsistent, which is impacting the duplication of efforts. It's possible that a researcher who never knows that such type of data is already existed will again duplicate the study and again put the money and efforts to generating the similar type of data which is already present. So there are also the challenges in the inability to perform the national scale meta-analysis, and also which limits the cross-institutional collaboration because nobody really knows that this type of research is already happening in this research group. So uh, this is making the animal phenotype data in institutional silos, and unlike of the genome data or other omics data, you can say that the journals also they don't really ask for submitting a phenotype data under some standard repositories because internationally also we don't have the minimum standards for the animal phenotype data. We have guidelines of FAO for the livestock and other domestic animals, but like that of other omics data, the guide, the checklist, or you can say the international standard is still lacking. So majority of that data is non-standardized, and you can say that it may have redundant trait names and other they vary with different measurements or protocols, and this data cannot be reliably pooled, and it is very difficult for to compare these data sets together, and uh, machine learning models trained on this type of data will not give any reliable results. So this is also due to because we don't have a metadata harmonization. When we want our data to be used in certain way, we will have to have a standard metadata format so that all of the data that is submitted are in a similar format with all the metadata fields that is required to understand that data. So it's very important that we have metadata standards. Until now, these standards are lacking. So many of the data sets lack the structural metadata fields. The environmental descriptions are missing. The breed meanings are inconsistent. So it's important. It is impacting that the genotype-phenotype integration becomes unreliable, and the reproducibility declines, and the fair compliance is limited. So phenotype without a strong metadata is biologically uh, incomplete. So coming to the last, that when we don't know where exactly is the data, it is very difficult to make that data interoperable across different systems. So to make that, we need to have data together and in a format which makes the data interoperable so that we can connect the different data types together to generate more of the information. So the bigger picture here for livestock data is all of the data from Indian biological data ecosystem on livestock research will come to the IBDC, which will allow it standardization, interoperability, and allow users to reuse, and will provide unique accessions, which will again improve the livestock health and productivity, and ultimately contribute to the One Health mission. So with this aim, or you can say vision in mind, we have developed the Indian Phenome Repository, which is basically a collection of different repository, or you can say the sister repositories. So initially we have started with Indian Crop Phenome Database, which is an active portal. And if you want to learn about the Indian Crop Phenome Database, you will find its webinar on our YouTube channel. So this IPR is act as an umbrella for these four databases. So currently ICPD and IAPD are functional, and very soon we will launch the Indian Human Phenotype Database also, and then we will move towards the microbial phenotype. So this allows this IPR allows the standard phenotype archival and sustainable archival from the Indian community, and our indigenous data on phenotype will be at a single place. So coming to the Indian Animal Phenome Database, you will find it as a user-friendly single solution for the structured phenotype data submission. We will give you persistent and unique IBDC accessions, and it will make your data fair, compliant, and you can able to manage your data, all the phenotype data, at the data in your account. So it will standardize your data. It will give your accessions to each study data and project. It will allow you to share data, collaborate, and build capacity. And we will also support the biotechnology-led different research projects on leading genomics and phenomics. So we will again follow FAIR, and all that data that is submitted in IAPD will be under CCBY and CNSA license, which is a very strict license, and so nobody can really use your data without the permission of yours. So this is the overview of the metadata that is used in IAPD. The hierarchy is project. A project can have multiple studies, and each study would have a data file and associated data. So you can submit your single project with multiple studies. You can submit your collaborative projects on single species. The consortia project submission can be done. You can also submit your multi-species project in IUPD also. So this is the basic submission and overview, which start with whenever a new user comes to IAPD, he has to register, then log in, and then follow registration, project registration, study registration, download the template, fill the sample information, upload the data, and again get the accessions. So any type of phenotypic data, be it the manually collected or the high throughput phenotypic data can be submitted, and it will allow you to comprehensively document all the metadata and also the sample attributes, the genotype passport data, replicate information in a template-based submission format. All our submissions are enabled, or you can say supported by animal trait ontology and tissue and stage ontology from Uberon, and the measurement ontologies from OBI. So this will make us each submission is not just a submission, we are curating your data. So starting with the, this is just an overview, and Isha will give you the detailed demo about how to submit data at IUPD. Starting with just account registration, you have to log in, then you will get your dashboard where you can start your project registration and study registration. As you can see that here in registration, your project will be connected with the CBI taxonomies and the publication BUIs. Here we are enabling ontology terms, then you can add sample attributes as of your choice, and then lastly, you have to populate the data and upload, and you can also upload the associated submission file. So this is the IAPD homepage, and then to get started, you can click here on get started. It will lead to the register page for this IPR, and then you can register on through this form. It is a very minimal form which will ask you for your organization, designation, country, and org ID. After signing up, you can directly log in. You will receive a email of confirmation, and then you can log in onto your account. So after login, you will see that it will allows you to select which database you want to go. So for animal, I want to go here, And this user dashboard will show you all the list of all the projects, studies that you have already submitted, what all requests and issues you have recorded. The left panel will show you tools like submit data, help, where you can see the SOP. You can also request us for deletion of records in case you have done anything incorrectly or by mistake. Then you will also find a FAQ section here and a discussion forum where you can create the request regarding the suggestions or any technical issue. You can read the data policy from here for ICPD and also for IAPD, and you can see your data at this page, and you will find that here is an advanced search panel. So for future use, when your data becomes too bulky, you can search your records from here. So going back to the submission, so when you want to submit a new data in Animal Phenome Database, it will guide you through different steps here. The first step is you have to register a project. So when you try to register a project, you have to fill information about your title, grant number, funding agency, and a brief description about the project. You will find all the help sections in these help boxes. So you can also give the DOI or PNID numbers of your publications which are related to the projects. Then you can select the which data access mode you wanted. Basically, when you want your data to be open access, you will select open access. When managed access is basically when you want your data to be accessed through request and not under open access. And the third mode is very for only selected or very critical data for no access, which anybody will don't know about which this type of data are really archived at IBDC. You can select the release date, and then for managed access as well as for no access, you have to give us a proper justification why really you want this data to be under managed access or the no access domain. You have to share the data sharing plan that when you want to submit or you can share your data or you can open access domain and how really you want to share your data. So then you can select whether your project is an individual project or a consortium. So when you click on consortium, it will give you fields to add your collaborators. You can add multiple collaborators and as per the project demand. Then for studies, because most of the animal studies require ethical clearance, and so when you click here, you can upload the ethical clearance document. Otherwise, you can go ahead with the submission. Then you have to provide the scientific name of the your biological system in which you have generated the phenotypic data, and automatically when you type here the taxon ID automatically fetched up from Insidium. You can add multiple organisms here, and you can always add how many organisms are there. Then you have to check box this box, which is basically for you to read the biotech guidelines and the protocol so that you can able to understand the data access and submission guidelines from the Department of Biotechnology. So then after filling in this form, you have to register project, and after that your project will be registered and you move to the second step, which is the registration of the study. So when you want to register a study, it's possible that your project can have multiple studies based on different meta traits. The hierarchy is such, for example, you are analyzing or screening a population of cattle for milk production or for heat tolerance. So these are two different meta traits, and based on that, you can study multiple studies, register multiple studies within a project. So you have to select the project. So as soon as your project got registered, it will be shown up here in the list. And after that, for example, if I'm adding this, automatically the organism registered in that project will be listed here. You have to provide a study title, a proper description of your study so that whosoever will in future uh, comes to your study and want to reuse your study will have a clear picture in mind that how the study is conducted, and we will have the complete idea how to use that data. So, for example, you have to just because our trait, tissue, and stages are connected with the ontology, you can add, and then automatically the ontologies for the traits will be showing up. Then you have to provide the start and end dates, and for example, the trait of your choice is not present in our database. So you can straightforward add a trait. You just have to provide the trait name and give it a description, appropriate description, and then you can enter the organism name in for which you are registering that trait, and then click save trait, and automatically the list got updated and you go ahead with your submission. For example, then you have to provide the start and end date of your study, then you have to provide the study type. We have an exhaustive list of study types, so you can select accordingly. Then you have to enter the place where the study is conducted, and automatically the latitude and longitude will be selected. Then this section deals with the authors. So whosoever has contributed to the study, all the data generators, students, research scholars, you can add their names here. You can add multiple names here. And then you have to provide the details about the experimental conditions, what exactly is your experimental design, what temperature humidity parameters are used, is there any other environmental factor you can add, and then whether your study requires a treatment or it's just on control conditions. So if you can say, yes, my study requires treatment, it will ask you for the treatment details, where you can select whether case or control, then you can add treatment agent, the treatment group, whether it's control or placebo or any stress or any agent. Then you have to describe your treatment, then you can give the duration and dose of the treatment, how you have administered that treatment routine, etc. You can add multiple treatments from here. Now, the second last section is the most important one. Now, here you have to list down all the traits which you have observed for that phenotypic study. Automatically, you can select which type of trait category you have studied. You have to type the trait name. Similarly, when you type, automatically the ontology terms will be showing up. You have to add the tissue, developmental stage, all that is connected from ontologies. In case the tissue and developmental stage of your choice is not present in our database, you can always add by clicking this plus button. Then you have to specify the measurement method in detail, what protocol or the reference protocol you have used, what software you use for the analysis, what data processing method, what quality controls are used, and you can add multiple traits to a study. So when you add multiple traits, you can do that from here, this button, and you can always remove. Then, in case if it is also associated with some genetic marker or the genotype or the mutation type, you can add that information here. And after that, you click register study. So after successful registration, you go to the step 3 of the process, wherein you have to again choose the study from this list, and automatically you have to provide what all genotypes. This is basically the list of genotype, one genotype per line, and specify the number of replicates, biological as well as technical. You can select the sample attribute, what all other information about your genotypic samples are you want to specify. It can be about species, age, feed, and any other. So if you have some other to add, you can always add, and after that you have to generate template. When you generate template, for example, let me just select and show it you. For example, I am generating T1, T2, 3 biological replicates, 2 technical. Now just one example, and let's do this. Generate template. My template got generated, and it will show you this template, which is pre-filled with ontology terms and all. And you have to specify the sample length status and all the B1, T1 values. And then this step is done. So after populating your phenotypic data, you go to step 4, which is the upload CSV. You have to select the study in which you want to upload the data, choose the file and upload. And after successful upload, you will be redirected to step 5. So before going to step 5, here in the upload section, we will, when you submit or uploading your file, we will validate this for the content in the file through different validation mechanisms, so that if the file is changed in any way, it will not accept, and if it will have some missing values, it will not accept. So carefully fill that only the blank columns, cells, and not try not to disturb the format of the file. So after that, you go to step 5. In case you have any other associated data which is essential for this study to go together, you can again attach any number of associated data to this. If not, you can skip this step. And after that, you will see that your study will be listed here. You can see that project. As you can see that this project will have a single study on uh, uh, the balance, and it is under managed access. It will be released on this day. So when you see, you can see that all your project details will be available to you, along with the links to the organism taxonomy, and also it will show you through a little knowledge graph that how many studies are there in the project. So you can always download that, and this will list you the what all associated studies are already there in this. So when you click on the study tab, you will find the metadata of the study, and you can always download it, share it with anybody you want, and this is how you can be able to uh, submit your phenotypic data. So as you can see here that every project will have a accession, and that is a persistent accession. It will not change during the course of time. So this way you can able to submit all your phenotypic data sets in a very reliable and easy way. For the users, we have the detailed SOP. You can go through these SOPs. It will guide you through the detailed process of submission. If you want to give us the feedback form is already integrated. You can give us the feedback also. So it will also guide you through your uh, FAQs if you have. So this is about the summary about the data submission process. Now after that, I just want to say that we have lots and lots of phenotypic data here in India. Unlike of that, uh, the sustainable archival frameworks for phenotypic data in India is just starting, and it's an initiative for your benefit, for the benefit of the researchers. So if you adopt their principles or the data submission in public repositories, it will definitely allows you to manage your research properly. So all these databases are there to support animal research here at IBDC, and we all know that we, as animal fraternity or the animal research groups, we generate lots and lots of data. But the times now are not just about data generation. The future of animal science will not be defined by how much data we generate, but it is by how effectively we make it fair, connected, and reusable. So IBDC is always there, the support team always there to help you out with your submissions. And apart from these resources, we have developed BioNode, which is a analysis platform, and it is very useful for the students who want to analyze their genomics and structural data. They can do by creating just account in simple ways. Then we, apart from that, we offer HPC support, and also we are ready to support the national mission programs. So with this I thank my IPR team. It's we are all girls team here at the Indian Phenome Repository Group. Isha, Nivedita, myself, and Vibha, who is our software developer, are uh, behind the IPR and IAPD. The ICPD, again, we have Mayuri, who is developing the Indian crop genome databases. So all that work you see are seen here is their efforts in making your research easy and fair. These are the core team behind the Indian Biological Database. You can see Professor Arvind Sahu, Dr. Debashish Mohanty, Dr. Dinesh Gupta, Professor Deepak Nayan, and Dr. Shashank Shekhar, the PIs of the project. And the acknowledgements are definitely due to Dr. Rajesh Tokre, Dr. Renu Swarup, Professor Siddhant Srivati, Dr. Satyakam Navi, Dr. Saurabh Raghuvanshi, who are our former lead, Dr. Richi Mahajan, Mr. Naveen Kumar, and the locus team who takes care of the HPC here at IBDC. These are our collaborators, the team at NIC, MII, DPT, RCB, and ICGB. So with this, I think IBDC is ready to support your research and please be the contributor towards data sustainability and conservation to revolutionize, to make advance the Indian biological data infrastructure. And with this, thank you. And this is just a feedback form in case you want to give us the feedback. Now I think we can check if there are any questions available. Okay, this is regarding uh, Prashantli is saying that if any e-certificate is available. Actually, for these type of webinars, the e-certificate is not available. These are just for your uh, information so that you can able to successfully submit data and know about that such and such resource are there at IPDC. So if you want any help regarding the data submission, we are always happy to guide you through that. So any other question, please, you can write to the chat box so that we can guide you guys accordingly. Most welcome, Kritanji. So with this, I think thank you all, and let's be now. Thank you so much. Bye bye.
Thanks, ma'am.
Okay, thank you. Bye bye.